Professor Vijay Yadav, thanks so much for joining me.
It is my pleasure to be with you today.
And thank you for putting up with our technological gremlins. I hear that you've had gremlins recording with large media companies previously, which makes me feel a bit better about myself.
Sure, I think it always happens. Don't worry. The technology is made by humans for our use, but it always glitches at the right moment when we need it.
As glitchy as we are. So today we're going to focus mostly on taurine, and I thought we'd begin with a series of rapid fire questions, which are notoriously difficult for scientists. I'd love it if you could try and keep your responses to a couple of sentences or less because we can get to the details later. But to start, what is taurine?
So taurine is an amino acid, is atypical one because it is having two different groups. It's an amino group, which is present in natural amino acids that we are aware of, and a sulfonyl group. And sulfonyl group is what it makes is atypical because in order to form amino acids to form a peptide bond, they need a carboxyl group. And because it has sulfonyl group, it cannot form a peptide bond. So it remains as a free molecule in to the most part in our cells.
And so it's not incorporated into proteins like those in skeletal muscle. Which foods are rich in taurine? And what are relatively typical taurine intakes for people with different dietary patterns? Maybe we could compare an omnivore to a vegan.
The taurine is coming from an animal-based diet only. It is absent in plants. So the pure vegan diet will not have any taurine in it. And the highest amount of taurine is present in shellfishes. So these shellfishes have like 1 milligram per gram of taurine. What it means is that even if you consume 500 grams of shellfish, you will get 500 milligram of taurine. And we'll come later in our conversation why I think it is not sufficient.
How do people's blood levels of taurine typically change with age?
Taurine is very interesting molecule. It is produced in adult life in our bodies because liver, for example, is a major producer of taurine, right? Liver can produce taurine. But if you look at the lifespan of a human being, the taurine production and its utilization changes. So in gestation, when we are in mother's womb, we do not produce taurine. And all the taurine, or at least most of the taurine, is coming from mother's blood through placenta and delivered free of cost to us. When we are born, a human being, when he is born, we still do not have the ability to produce taurine to any significant extent. And that point, it is coming from mother's milk. That is where the concept of essential, I personally think that taurine is a vitamin during gestation, during early human life, and becomes semi-essential because we get little ability to produce taurine from liver and other organs. So it becomes semi-essential during adult life. But in later part of our life, we start having compromise in taurine production as well as its utilization. There is taurine resistance that starts developing in our bodies. A similar phenomenon you can see in obesity, for example. You get insulin resistance. Your body has insulin, but you're not responding to insulin. So during aging, our taurine abundance and its activity goes down. So you have a period where it is a vitamin. I classify taurine as a vitamin in early life. In midlife, it becomes semi-essential. And in the later part of our life, it becomes essential again. That means it becomes like a vitamin again. So we need to view nutrients very differently when it comes to taurine intake in human being. Our taurine intake during adult life is sufficient to the most part because we are able to produce some taurine by ourselves. Our cells are healthy. but it is not sufficient, neither by our body's production ability or by intake during late life, because we get a problem in absorption, problem in production, and problem in taurine activity.
You're definitely a scientist. That was a long answer for a rapid
Oh,
fire
I'm so
round, but
sorry.
it was an interesting answer. We might come back to it, but it's particularly interesting that there could be taurine resistance if you compare it to, say, insulin resistance, because insulin resistance or leptin resistance is typically in the face of elevated levels of those hormones. Whereas I think what you're suggesting is that taurine synthesis might be going down and cells could be becoming more resistant to it, which is distinct from many other molecules that we might consider. But back to rapid fire questions. Can taurine make mice live longer?
Yes, the short answer is yes.
Can taurine make worms live longer?
Yes.
What's the scaled up human equivalent dose from the lifespan experiments that you did on mice?
It is 3 or 6 gram taurine per day for a 80 kg human.
And is that dose in keeping with existing randomized controlled trials on taurine supplementation by adults?
So there are different trials that have been done, and they have done from 1.5 grams per day to 12 grams per day.
And what are some of the beneficial effects of taurine supplementation shown by those existing clinical trials?
So taurine has been shown to reduce obesity in humans, increase exercise capacity in humans or muscle function in humans, reduce anxiety in humans, alleviate post-menopausal symptoms in humans. It has also been shown to improve eyesight in humans, skin health in humans, reduce liver damage in humans, um, decrease insulin resistance in humans, or have a better glycemic control. I have come across a plethora of clinical trials that have tested taurine in specific diseases, such as menopause or obesity, diabetes, retinal degeneration, or skin health, and so on and so forth.
I hadn't seen those studies on eye health or on postmenopausal or menopausal symptoms. So look forward to digging those out in due course. Is taurine supplementation safe for most people?
So taurine, according to European Food Safety Authority, it is safe up to 6 gram taurine per day.
Have your thoughts on roles of taurine in aging changed much since the publication of your paper in Science last year?
It has become more grounded, in my view. It has become more acceptable in terms of using taurine as an intervention to increase human healthy lifespan.
There's a geroprotective agent, so it's something that is countering processes that are involved in aging. How promising do you think taurine is relative to other in vogue candidate geroprotectors, such as rapamycin, NAD precursors, metformin, senolytics, the list goes on?
I personally view any natural molecule to be having a stronger effect in a long run, because natural molecules tend to have little or no side effects if used in appropriate doses. Having said that, synthetic molecules also have their benefits. Um, and everybody has their favorite molecule. But when we talk about aging, I do not think anybody should look at one molecule in particular with a narrow focus. Because if you start looking at narrow focus, you start doing propagandas. So at the present time, I do not want to have a favorite molecule. Be it a taurine, rapamycin, senolytics, metformin. It can be anything, or it can be a basket full of pills that we may take 10 or 20 years down the line to increase healthy lifespan. It doesn't matter to me, because I think we need to study more of these molecules in order for us to alleviate any side effects if they have one. None of these molecules have been tested in human clinical trials for aging as an indication. So we will not have that answer. The question that you raised, we do not have an answer because none of these molecules have been tested yet.
Pleased to hear that because so many scientists seem to get quite wedded to their pet substances, whether that's nicotinamide riboside or rapamycin. And I completely agree that different candidates are going to be better suited for different individuals, depending on what they're facing, their genetics, their goals, their existing health conditions, and so on. But if we move on from the not very rapid fire round to some background to taurine, one of the things that you mentioned is you think that whether taurine is essential might fluctuate over the course of the lifespan. And you use this term semi-essential. I'm more familiar historically with the term conditionally essential, which is often used in reference to various different amino acids. Take glycine as an example, with the suggestion being that there are specific circumstances in which it can become essential because the body isn't able to synthesize enough of it to meet metabolic requirements. Is your use of semi-essential synonymous with conditionally essential, or is it distinct?
I think is slightly different. When you talk about conditionally essential, for example, if somebody is having a liver disease or liver is not able to function very well, they will have less taurine production. That is conditionally essential. Where there's a liver disease, you are not able to produce it enough in adult life, and then you need taurine, right? But when you look at semi-essential, your body is able to produce taurine to a limited amount, but you need it exogenously for perfect functioning without having any underlying disease state or organ dysfunction. So that becomes semi-essential.
This conversation reminds me of a theory that was put forward by the recently late Bruce Ames. He put forward his triage theory. The crux of it is that during times of insufficient intakes, the body is going to prioritize diverting resources to short-term survival rather than investing in maintenance processes that are more relevant to aging. And in that paper, he listed various different dietary compounds that might fit into this category, and taurine was one of them. And I was wondering whether that particular theory has shaped your perspective much, and what some examples of other dietary constituents might be that would fit into that category.
So that will be a very long answer. I will
Okay.
warn you first. Um, so in any of these nutrients or dietary changes or molecules required for our diet, we need to look at human history. And human history is very long, right? It can see millions of years, right? We have been evolving somewhere in Africa. There were changes in African subcontinent where people now had to move out. The human beings and our ancestors had to move out of Africa to get better food access, better resources, better place to live, because the population grew, right? They needed more resources. And this changed the our ability to really utilize the resources around us. And many events happened during the course of this migration history. For example, invention of stone tools, invention of fire, for example. These two were very critical for human evolution. Invention of stone tools allowed us to break open a skull and get nutrient-rich fat brain of an animal. We also utilize fire. Fire allowed us to cook tubers, such as which are underground, like potato or whatever kind of potato at that time was available. And these tubers were carbohydrate rich, but they were not accessible as a nutrient to us. Fire allowed us to cook them, to break it down into smaller pieces, and that allowed us to have a better ability to absorb those nutrients in our bodies. So, invention of stone tools, invention of fire really dramatically shaped the ability of humans to grow. Our brain grew bigger in size because we had now better access to nutrients. A more dramatic event happened later on, not long ago in human history. Modern humans, like history could be looked into like 200,000 years. But a major dramatic event that happened around 10,000 years ago, which is invention of agriculture, somewhere in Iraq. And this invention of agriculture, in my personal opinion, although it allowed us to have better access to food and resources, was detrimental for human health. So invention of agriculture made us self-domesticate ourselves. When I talk about self-domestication, it means we were living in wild, hunting animals, gathering tubers or eating berries or fruits, whatever we had access to. But when we became a agricultural society, we started growing our own crops. We brought a lot of animals from wild to domestic, and they domesticated with us, such as cats, dogs, cows, horses, and so on and so forth. And this self-domestication of ourselves and domestication of other species led us to face a nutrient imbalance because now our food became more carbohydrate rich. Many diseases precipitated or cellular dysfunction precipitated, but we did not face any consequences of those cellular dysfunctions because we were 100 years ago, our average lifespan was 30, 35 years. We didn't face it, right? But we started slowly and slowly because of the invention of modern technologies, medical care. We started to live longer. And that is when many of the micronutrients, you can call them, semi-essential nutrients, you can call them, or nutrient imbalances became apparent. So vitamins were discovered starting in 1920s. So it tells you something more fundamental to our health. Like vitamin, it started with vitamin B's, vitamin C, many diseases like scurvy, beriberi. You can pernicious anemia caused by B12 deficiency, for example. So these discovery of these vitamins just started 100 years ago now, right? So that tells us that many nutrients are still unexplored, or we do not yet understand our nutrient composition, which is ideal for our health. So no nutritionist can say that this nutrient composition and ideal nutrient composition is for you, be it a vegan diet, keto diet, or any diet that people prescribe, because that human genetics and nutrients have co-evolved together. Many genes have been lost in populations which stopped using particular food. For example, if you look at many Asian populations, Southeast Asian population, enzyme lactase is lost because they were not consuming milk anymore, right? But it's present in other populations. It tells you that genetics and nutrients need to be looked on side by side when you are talking about nutrient changes that we want to incorporate in our life to live longer and healthier. I don't care about living longer, I care about living healthier so that we face less more diseases at the end of our lives.
I made lots of notes while you were speaking then. So I'll try and tie a few threads together. I agree that our nutrition, our health deteriorated with the advent of farming. And I think that we see that in fossil records. And I thought it was interesting that you said that we were domesticated, because I know that some people have argued that it was really the crops that domesticated us, which flips a lot of people's ideas on their head. But I think that we see that evolution of shallow adaptations to various different nutritional changes. You mentioned lactase persistence, that being one of them. There are many others, alpha amylase copy numbers and their relationship to starch intake, enzymes involved in desaturating fatty acids, and so on. Based on what you were just saying, I was wondering, would your hypothesis be that groups of people who historically have had relatively low taurine intakes, let's say they've consumed small amounts of animal foods and in particular of seafoods, would synthesize greater amounts of taurine, and hence their taurine needs might be lower than people from, say, higher latitudes who have had a more meat and fish rich diet and have relied less on plants.
Possible, but being a scientist, I would say that unless we investigate those correlations, we would not have an answer to it. It's possible, very much possible. There's one thought that has been running in my own mind for last decade almost now, because if a pure vegan diet is having less taurine in it, perhaps our body is able to produce taurine better, or there's another possibility exists that there are taurine mimetics present in vegetarian diets, which can do the same function as taurine does.
And taurine synthesis requires pyridoxine, vitamin B6.
So again, it is not very well understood. There are many enzymes in this pathway.
Classes
Cysteine.
are cysteine.
Yeah, you can say in the body, if you're producing, it comes from cysteine. And it can also come from methionine. We described a pathway of vitamin B12 dependent synthesis of taurine almost a decade ago now, 2014. Paper published was in Journal of Clinical Investigation. So there are many pathways that are coming to produce taurine, and this is not well understood till date. What is the entire cellular machinery which is required for taurine production?
Watch this space. It makes sense to take a step back and ask you how you came to taurine in the first place. What piqued your curiosity?
So before I came to taurine, a lot of people came into taurine world. And the introduction of taurine in society has followed a very dwindling path. Um, so taurine was discovered in 1827, right, in ox bile. But molecular biology and biochemistry was still evolving for almost 150 years. So till about 1950s, it remained in oblivion. Um, and in 1950s, society became rich after World War II, and society had a lot of pets like cats and dogs. So we needed a pet food to be given to our pets when to go to 9 to 5 jobs and go to 9 o'clock in the morning, put some pellets in our bowl of a pet, and they're happy when we come back home. This introduction of pet food in the society led to precipitation of many diseases in pets, such as cardiovascular abnormalities, obesity, growth retardation, hair loss. The most striking aspect of this pet food introduction in society was that cats went blind. And this blindness in cats was very worrisome. It's a very difficult sight for a pet owner to see himself that his cat is blind, right? So this increased interest from society to fund scientists to figure out, okay, what is going on in the cats? Why are they blind? This took around from 1950s, it took around 15, 17 years, and it was discovered in 1975 in a seminal paper published in Science magazine that showed that retinal degeneration or blindness in cats is caused by one molecule deficiency in their diet, and that was taurine. Another decade later, another paper was published that showed cardiac failure or cardiac abnormalities in dogs are also caused by taurine deficiency in their diet. Again, the paper was published in 1987 in Science. So these studies, along with hundreds of other studies, okay, have told us that taurine levels in the blood is associated with the health of a human being as well as animals. But when we were starting, we were not looking at taurine as a molecule at all. We were trying to look at, okay, what happens in the during aging in a human being in the blood, trying to identify, okay, what molecules are changing? Is small molecules, proteins, or lipids, or what is the dramatic change through metabolomic approaches which is happening in the bloodstream of compared to a young human to an old human? And this screen identified that there were many molecules going up and down, and 99% of those molecules are still under investigation in my lab. One molecule that actually went dramatically down in aged population, and I'm talking about human beings right now, was taurine. When one of my senior scientists came to me and said, do you know what this molecule taurine is and what does it do? In 2011, that time I was in Cambridge, in England, my answer to her was, I have heard about the name in some energy drinks and things like that, but I barely know much about it. Let us find out. And this actually was a initial curiosity from her and from me. We put a team of investigators to really figure out, okay, what does taurine do during aging? And we took a approach where we started supplementing mice with taurine, middle-aged mice. When they have they before the taurine levels started declining, we started supplementing them. It reversed their deficiency and it made them live longer. Not only it made them live longer, it made them live healthier. They were healthier. They were looking at brain function, reduced anxiety, more memory, more muscle health, more skin health, more bone density, better liver function. You can name an organ, better glycemic index. You can look at all the functions. We literally created a health clinic in a mouse to really show that not only these animals are living longer, females were living 12% longer, males were living 10% longer. Not only they are living longer, they are also living healthier. And not only mice were living longer and healthier, worms were living longer and healthier. We did many experiments in zebrafish, they were becoming healthier. We did the experiments in monkeys, they were living healthier. So these studies tell us that, okay, taurine as a molecule is only a one part of a puzzle of what nutrients are required for our proper health. It is not the entire book. It is just one page in our book of nutrients required. And we do not understand, I underscore, we do not understand what nutrients are required for proper health. Are what we are eating in the morning, is it healthy or not? I do not think that it is healthy because we don't know what do we need. Unless you know what you need, you cannot get it because you don't know where to get it from.
We will come back to those experiments you just mentioned because they form the body of the paper that was published in Science. And it seems that Science has a rich history of publishing important research on taurine, going by your description of the history of the science of taurine. But just returning to something that you said then, human levels decline with age. And I think they can be more than 80% lower in elderly adults compared to their younger counterparts. What do you think is driving that? So simplistically, that could be due to several factors. It could be that people are just consuming less taurine, which frankly seems relatively unlikely. It could be that people are synthesizing less of it. It could be that people are excreting more of it. But it could also be that taurine is being shuttled between different tissue compartments differently. So it could be there's more in the blood, but there's less in some other tissues if we consider blood to be a tissue. What's your speculation about what's going on there?
Well, you answered the question actually already. There's a combination of all these factors: less absorption, less production, and less activity of taurine in the cells. So it is not able to do its job very well because there is less availability of it to the cells to do the function properly.
Do we know much about how levels in the blood relate to levels in different tissues. Example that comes to mind, which would be quite easy to study, I imagine, would be taking muscle biopsy and then looking at the taurine concentration per unit of mass of particular type of muscle fiber. So take type 1 muscle fibers from the lower leg and then compare those fibers in people of very different ages and look at whether the taurine concentration differs between those people. Do you know much about how blood levels relate to tissue levels and whether that fluctuates across the lifespan?
So one has to look at now which tissues produce taurine and which don't. So liver levels decline slightly during aging. Muscle levels decline slightly during aging. Muscle does not produce taurine. It has zero ability to produce taurine. So whatever taurine is present in the muscle is actually coming from the bloodstream. It's possible that muscle, which is one of the largest organ in the body, is a mop,
Yeah.
quenching all the taurine to itself during aging. And so many organs which have no ability to produce taurine need it for their function. The highest amount of taurine is present in muscle, in liver, in brain. And many organs don't produce, neurons don't produce it. There are some cell types can produce in brain, but not all. So what it tells you is that the cells that don't produce taurine at all, they need it for their function. Skin levels go down. Skin can produce a little bit of taurine, but it goes down with age. Okay, eye levels go down with age. Depending on which organ people have looked at and which species people have looked at, the answer can be very different.
And the lower liver levels suggest that synthesis is likely blunted because most of taurine synthesis takes place in that tissue. Do you know much about the density of taurine transporters in different tissues? I imagine that it varies quite a lot.
Yeah, muscle is the one of the strongest expressor of taurine. It is ubiquitously expressed. Short answer is ubiquitously expressed, and cell types that don't produce it on its own, they have a higher level of taurine transporter. Now, when I say taurine transporter, a generic knowledge tells you that there's only one transporter. A scientific knowledge tells you, no, there are at least 4 or 5 transporters. One of them is a major one, like which is known as SLC6A6 approach, the scientific name for that, or TOUT, they will call it as well. But there are many other transporters required to, to import taurine within the cells. In the GI tract, we have a different transporter, which allows us to absorb taurine. And so therefore, there are the taurine transporters are many taurine transporters are present in the body and they perform different function depending on the cell type that you're looking at. Some of them need a lot of taurine to import taurine in the cell. Some of them are very sensitive. They have very small amount of taurine present on surface, like taurine transporter. It is able to take it inside. So we have to be a little bit conscious of the fact that there are many proteins that can do the job depending on the cell type and condition.
Do those transporters seem to relate to taurine requirements or turnover? So, for example, if someone was doing a lot of exercise and there was a lot of contractile activity in their skeletal muscle, then could that increase the density of taurine transporters, similar to how we might see changes in GLUT4 activity on skeletal muscle fibers to shuttle more glucose into muscle? And then related to what you're just saying, do taurine's metabolites have distinct transporters, or is that not a relevant factor?
Could you repeat the last part?
So, do different taurine metabolites, like hypotaurine,
Mhm.
have their own discrete transporters, or are they interconverted back to taurine and then taurine itself crosses through transporters?
You mentioned a very important thing, like people who do exercise, do they have more taurine levels in the muscle? Or it's very interesting. If people who exercise is one of the stimulator of taurine in the blood.
Mm.
So if you have a baseline, a level of taurine in your body, and 1 hour after exercise, taurine levels go up in the blood. Exercise being a healthy state of a human being, right, is one of the best known mechanism by which we can delay our aging or increase our health. It tells you that taurine levels may be doing something to exercised individuals. It may be communicating to different health. We feel better after exercise, right? Our brain feels happy, our muscles are all geared up. Um, and any one organ cannot be looked in isolation because all our organs are communicating with each other. The concept of endocrinology tells you that many organs are secreting factors which are acting on a distant organ, and that is hormone, right? So we have this plethora of changes happening in the body, which are directly related to a particular activity, in this case, exercise. So possibly many of the health benefits of exercise could be mediated by high taurine abundance in the blood. Where the taurine is coming from, nobody knows yet.
Brings to mind so-called myokines, doesn't it? These contraction induced changes in cytokines produced by skeletal muscles that then have remote effects on various tissues, including the brain. Am I right in thinking that taurine levels in the blood vary quite a lot between species? And in your science paper, you mostly studied C57 black 6 mice and rhesus monkeys, macaques. How do their levels compare to the levels in human blood?
The answer to that question is more complex. Because in order for us to be able to compare across species, we need to assess the levels on the same platform, right? And that has not been done. Okay. So we looked at taurine levels in the bloodstream by HPLC. We looked at taurine levels in other species by ELISA. So ELISA does a qualitative comparisons. It's quantitative, but depending on the antibodies being used, the levels can vary dramatically. And human beings, it is in micromolar range. Um, if you go to lower species, it can be higher. So, and that also tells you about the volume of the blood that they may have, or volume of the extracellular fluid they may have, like if you look at C. elegans. So the values vary dramatically between species because every species has a different requirement of taurine for their functions.
And does there seem to be any relationship between the amount of taurine in an animal's habitual diet and their blood levels? So if you take mice and macaques, they consume less meat and seafood than us humans. And so one might imagine that they might synthesize a little bit more taurine and rely less on their diet for intake. Do you think that the relative effects of taurine are likely to vary a lot between these different animals?
Of the same species?
No, of different species. I read one critique of your study, and being frank, it felt like the writings of people who liked being contrarian. I don't think that all of the arguments in it held water, but the gist of it was that they think that the effects in mice are going to be inflated relative to humans, in part because of differences in synthesis, which I would extrapolate might relate to intakes
Mhm.
and intakes over their evolutionary history. So I was wondering what your response to that suggestion would be.
Sure. I think all the criticism needs to be looked into, especially if this is coming from a well-informed person. be it a layman, or be it a YouTube influencer, or be it a scientist. It doesn't matter where the information is coming from.
Mm,
So yes, many of the effects could be higher in mice because you have more controlled environments, same genetic background. These are inbred animals, right?
mm,
They're not outbred animals. Then, and that is why you need to study that in many model organisms. This paper took 13 years for us to accomplish. It is easy for somebody to read for an hour and comment on it.
mm,
We started this study in 2011. We got our mouse data in 2016 that it was making mice live longer and healthier. We did not stop there. I did not believe that a single model organism can answer all the questions. We then went back to worms. We saw the same effect. In fact, worm effect was larger. It was up to 30% larger, increasing lifespan. We did not stop there. We went to zebrafish. We see the same effect. We did not stop there. I wrote down a study protocol. I moved from Cambridge to New Delhi in India to do a monkey intervention study. It took us 5 years to accomplish monkey intervention to show that if you supplement once a day taurine to monkeys, it makes them live healthier. The many of the health matrices improved. We did not stop there. We went to human beings. We looked at the 12,000, close to 12,000 people from Epic Norfolk cohort and looked at their longitudinal health data, looked at their taurine, hypotaurine, N-acetyl taurine, other metabolites, and correlated that to the clinical characteristic of those subjects. And we then showed that people who had low taurine levels had poor health. So his or her criticism is valid. However, one has to read the paper in its entirety in order to make an informed decision about the validity and robustness of the findings and what are we saying. Even after all these species were tested, we say at the end, the last sentence tells you in the paper that a clinical trial is a must to see whether taurine intervention will delay the pace of aging. And I am very specific in stating that. Anybody else would have gone haywire. There are 56 authors on this paper, people from expertise in worms, in zebrafish, in C. elegans, in yeast, in monkeys, in health measurements, clinicians. So we all have come up with a conclusion that yes, fine, all these studies are fantastic. We are seeing a positive effect in a variety of species, but we need to wait for the clinical trials to be accomplished before going out to the wider public to say that, okay, it is safe now. You can go ahead and take taurine. It will improve your health, will make you live healthier and longer.
It's very prudent of you. And also, just a comment on the author's piece. They seem to completely disregard the existing clinical research on taurine, which maybe isn't a home run, but it's consistently very positive. And what I find funny is that they could pick apart a paper like yours, which is very thorough, and there are various things within the paper which we might get to, which point to the quality of the data, but also be very optimistic about some interventions that have scarcely been studied in humans outside of a very narrow range of clinical populations. For me, that doesn't make sense. It just highlights the flaws in their thinking. But just to flag an example of the high quality experiments that I was alluding to, if we look at your experiments on mouse lifespan, you mentioned that both male and female mice live longer when they were supplemented with taurine beginning in middle age. And the increase was 10 to 12% in median average lifespan, which is on par with many of the better interventions or more efficacious interventions that have been studied to date. If you look at the control group in your experiment, then they were quite long-lived animals. Brian Kennedy is a very good scientist. You probably know him personally. Put forward the 900-day rule not that long ago, suggesting that if you look at the control group in an experiment done on mice and they live much shorter than 900 days, then it's not that you can disregard the data, but the effect sizes of the intervention are likely to be largely inflated.
Yes.
Whereas with you, that clearly isn't the case. One of the things that I was wondering is whether you are putting taurine forward for inclusion in the interventions testing program. A lot of people speak about this as the so-called gold standard in the development of candidate gerotherapeutics, and you can probably describe what the ITP is better than I can. But building on those experiments that you did on mice showing that lifespan extension, do you have any plans to submit it to the ITP?
We had a discussion with some investigators who were part of ITP, but ITP has a limitation. They only do interventions in pellets.
Okay.
So now we have to take into consideration, okay, how are we achieving the dose? We ourselves in the beginning, it's not part of the paper, we did continuous administration of taurine by putting in the diet. And we put a single shot administration by going through gavage. So when you are elevating taurine in a one shot, it is going to go into the tissues more strongly because you are over flooding the transporter present on different tissues to be taking up taurine rising in the bloodstream. But when you are increasing taurine slowly, it is not going in. When we analyze, for example, this short the in a 3 months study we did in the beginning, where we did the continuous administration and single dose administration per day, we saw that organ health in different organs was much more improved compared to when we did the continuous administration. So this founded the basic. These pilot experiments are not part of the paper. It's present in supplement somewhere.
Mm.
So this founded the basis where we then went ahead and single dose. It is a cumbersome experiment for students and postdocs to do. It is much more labor intensive, where because you need to go every day, feed 400 animals, either vehicle or taurine or any other molecule in this case, and then analyze them for almost 2 years. You're doing that job, Monday or Saturday or Sunday, it doesn't matter to you. Right? We did alternate ourselves to do those experiments. I did myself gavage going into the weekends. So what? Um, so yes, we had discussions, but it has not come into fruition because it is the inherent limitation of ITP program to be able to test molecules where the data is available, where people have tested a single dose administration versus continuous administration. And this is not only true for taurine. If you look at other molecules like PTH, for example, parathyroid hormone, I'll give you an example. If you do intermittent PTH administration, then only it improves bone density. In humans, I'm talking about. So there are other, I do not think that one should classify as any program. Those are also scientists who are doing in their laboratories. This is just a
Mm.
program being run in 4 places. And in this study, the experiments were done across the globe in different, even worm experiments were done in 3 different labs. In Arnav Mukhopadhyay's lab in New Delhi in India, in Matt Kaberlein's lab in Washington, and here in Georgia. So we have done multiple different experiments in different laboratories. Yes, sure. If somebody wants to do once daily administration of taurine in ITP program, I'm happy to discuss them. But there was no positive outcome when we had discussions with few investigators part of ITP.
It makes me think of my response when they tested some senolytics, because they published their work on fisetin not that long ago. And when you look at senolytic interventions, fisetin, quercetin plus dasatinib, whatever it might be, the purported mechanism involves this hit and run where you want a high dose for a brief period and then those senescent cells are cleared, but you need the bolus. And just adding it to chow might never achieve the peak levels that are needed to actually produce that senolysis. And similarly with taurine, if you need to spike levels in the blood high enough, then just adding it to the chow, you're never going to reach those peak levels. Another interesting comparison will be looking at skeletal muscle protein synthesis. There seems to be this leucine threshold that you need to hit on a per meal or per snack basis to maximally stimulate it. And if you never get past a 3 gram of leucine bolus, then you might not get that. So I hadn't thought about that in your context, but that makes complete sense to me.
To be just, ITP is a great program for
Yeah.
any of the
Oh, yeah.
interventions when it can be done through the mechanisms that they have in place.
Yeah.
Um, and maybe taurine, we have not tested taurine for lifespan studies given in the pellet diets. Maybe it will
Mm.
do the same job in a long term. We only did for 3 months, right? For a
Mhm.
single shot administration versus continuous administration. Maybe it was
Mm.
going to do the maybe better job of 12 gram, sorry, of 12% lifespan increase, the median lifespan. Maybe it will do better job. I don't know. But somebody has to do those experiments. We are planning to do those experiments ourselves as well. Get the pellet diet, put some 240 or 300 mice on the diet for a couple of years and see what happens. But we need some funds to really do those studies. Taurine study has never been funded by any government agency till date. Only last year we got some funding from NIH to do extension of some studies to for taurine. It has never been funded. I think it tells you a lot about what kind of things can get funded as well. We need money to do the job.
I'm wondering what you're alluding to there. Taurine being a natural compound present in foods is not something you can necessarily patent. So
Yeah, no, not
it's not,
necessarily to make a lot of money for industry.
yeah, okay, I understand. If we look at those experiments you did on lifespan, then the extension of life was slightly greater in female mice than male mice. Both did live longer though. And then the follow-up experiments that you did looking at your health span metrics specifically used female mice. Would you expect similar effects in males? And why did you choose females? Was it because the lifespan extension was greater in females, or was there something else at play?
In the beginning, it was to reduce the cost because you
Mhm.
need to be able to do the a more intensive investigation across different organ spectrum.
Mm.
But to be just, we also did in males the health span measurements. It is not present in main figures. You have to go to the supplemental figures in order
Oh,
for you to see the health effects in male mice as well.
you did mention it in the paper, but obviously you focused on the females. I looked briefly at the supplementary material, but it was actually specifically to look at the exercise protocol that you used for humans, just to understand something about the test, which was a graded cycling test. But in general, the male mice responded relatively similarly. And you mentioned some of those health improvements that took place. So increased bone mass, changes in body composition, the taurine supplemented animals gained less weight and had lower percentage body fat. They had improved exercise performance, both in endurance exercise, but also a measure of strength endurance. They had improved memory in a spatial memory task and reduced anxiety, reduced proxies of depression. Obviously, can't really say that they were less depressed because we're speaking about mice here. So we've got to look at surrogate endpoints. They had faster intestinal transit time, suggesting that their function was more similar to younger animals. They had better blood sugar, they had better insulin action as well, and they had blood cell populations that look more similar to young animals too. So, as you mentioned earlier on, across the board, you saw substantial improvements in their health, almost irrespective of the tissues that you looked at. Were there any other things that you looked at that didn't make their way into the paper, or did you include everything in the paper? I realized that in this particular paper, you've done, I don't know how many experiments, but certainly dozens of experiments, which is often the case in papers that are published in Cell and Nature and Science. But was there anything else that you looked at and didn't report? And if so, for what reason?
No, we actually, I am of the opinion that we need to be transparent and put everything out there, let people. We did not see an effect in East, it is present in the paper. So we included everything that was done in the paper directly. In fact, uh, one of the discussions that we had internally, that whether it is worth to put the yeast data out in the literature in this paper, because yeast had no effect of taurine
Mm,
on their lifespan.
mhm,
Then the discussion we had among ourselves, mm, that I think it is important to put it out there, because otherwise somebody will spend a lot of public money to do investigation in the East and ends up in the same conclusion. So let us put it out there. It has no effect in East, increases lifespan in worms, mice, monkeys, health. We need to put it out. So we put everything out. Then a lot of information present in supplemental figures. In
mm,
male mice, the all the health matrices are present in figure S3, supplemental figure S3. It is in supplemental data sheet. Um, we also did a lot of studies in mice that do not have taurine transporter. They live, so there is like an induced taurine deficiency. They do not have the ability to take taurine in the cells, and they live shorter. They they live like average life, median lifespan was something like 17, 18 months compared to 28, 29 months of a normal mouse.
mm,
So we not only did a gain of function experiment where we put taurine in the aging animals, we also removed the taurine transporter to create induced taurine deficiency. And these animals live shorter. They had a variety of health deterioration parameters like low bone density, muscle wasting, more anxiety, memory, glucose intolerance, and so on and so forth. So to get these are present in supplemental figures. There are a lot of supplemental data in this paper than we could put in the main figures.
Tell us a bit more about the health span experiments that you did in the macaques. I didn't realize that you personally had gone over to New Delhi for that. Also, tangentially, I can only imagine what it's like working with macaques. Anyone who's spent some time around them knows that they're a massive pain in the ass. They're very entertaining, but they're a lot of work.
I learned that early on during in my scientific career. Um, I used to work with macaques in my PhD days.
Hmm.
Um, my PhD supervisor, I think, put a lasting impression on myself. His opinion was that you should always use the best model to investigate a question. And why did I end up studying macaques in my PhD was that we were trying to study how human menstrual cycles are regulated during pregnancy and non-fertile cycles. And we discussed over a period of 6 months during the initial years of my PhD. And he told me, I was not aware of it, of course. I was just a young graduate student. Only monkeys can be used as a model organisms to study human menstruation or effect on ovarian cyclicity. Because no other species on this planet undergoes menstruation except monkeys and humans. That is the evolutionary paradox. Why on earth the highest evolved species, at least we think that we are highest evolved,
Mm.
undergoes during the course of a cycle such an immense blood loss, which it has to regenerate again.
Mm.
Nobody has ever been able to explain that. So I used monkeys as a model during my PhD time to investigate this pregnancy versus non-fertile cycles. And it was each experiment used to last for 2 or 3 years. And that allowed me to appreciate the importance of animal model organisms to really investigate. Not only monkeys I use as a models, I use cows as a models and rats or mice as a model to understand how the evolution has shaped the physiology as far as the reproduction is concerned. Monkeys are one of the best animal species one can use to investigate any intervention, especially in the context of nutritional intervention. It becomes much more important because they have a similar physiology as us, right? We are providing them a controlled diet in this case. So in monkeys, what we did is we came up with the protocol. We fed them 250 milligram per kg body weight, which is equivalent to around 500,000 milligram per kg body weight in the mouse. Once a day, we made my PhD student that time. He made a banana paste, put taurine powder in it because you cannot give monkeys a glass of water they can drink. They cannot. They will not. First of all, they will throw it away. So put the banana paste. We put taurine in it, powder, mix it, made a roll with the bread, and then we gave them, and they ate it because it was sweet. And this was done as a first meal of the day, so they will not discard it because first meal of day was given around 10 or 11 a.m. in the morning. Now, at that point, we in the facility we were giving food like fruits and other things we were giving them. So I thought, okay, let us do it in the morning because they have not eaten, they have no food left over in their bowls. Give them something and they will consume. And we stayed in the room till monkeys consumed the food that we had given them. So we ensured that monkeys consume taurine per day to the amount that we were giving them. And we did this experiment for 6 months, once a day. And at the end of the 6 months period, we looked at the health metrics compared to the control animals which were given vehicle here. And we found that body weight was reduced. They had less fat gain. They had better bone density. They had a better glycemic index, less liver damage. Uh, Myeloid to lymphoid ratio was shifted towards a better immune system function. They had oxidative damage markers we measured in the blood, like lipid damage markers or protein damage markers or DNA damage markers. They were reduced in these monkeys. And this was challenging because monkey research is challenging because we now had to, in fact, these are things are not stated in the papers, right? We had to figure it out how to do the DEXA measurement of a monkey. And DEXA machines are costly. Now we had a wonderful, I think, I would say he was a great entrepreneur. I really like him. His name is not in the author list because I cannot put that in the author list. But he said, you can go ahead. We have a spare DEXA machine for humans. He runs a lot of series of clinics in Delhi and other places. We have a spare DEXA machine. Go ahead and use it. You just pay for the source of or the machine time cost. And we had to anesthetize monkeys, put them in the truck, transport them for DEXA measurements. Once they underwent DEXA measurements, they were transported back to the facility. So it is logistics were really something that I think I learned a lot in this, how to really put things together, how to try to talk to people to get things done to the minimal cost possible because we had no funding. And there was a great learning experience for myself as a scientist, as a maybe a project manager, you can call it, to really figure out a way to get things done. So these monkey experiments also told us that monkeys were living healthier. Of course, we cannot do lifespan measurements because these are monkeys live long life, like 15, 18 years on average. You cannot do lifespan measurements, but you can do a health measurements. And perhaps in the future, which if we can do it, I would like to do a lifespan study in monkeys. That would be on my chart rather than doing experiments more in monkeys with the lifespan and health span. I would do monkey intervention study if I can get enough funding to do to replicate the finding to in the closest species possible. Because at the end of the day, we use different species as model systems. These are just one model, two models, or three models: mice, zebrafish, C. elegans, yeast, Drosophila, monkeys. These are model organisms. We need to test it in as best model as possible. The best model will be human, but those really require a lot of funding and resources to be done. I'm sorry if I gave you a long answer than you hoped for, but I got excited and I remember my stressful days of figuring out how to do the DEXA measurements in monkeys.
That's great. You mentioned DNA damage there, and that, in a circuitous way, brings us to the hallmarks of aging, one of which is genomic instability. And these hallmarks of aging were put forward over a decade ago now. And initially, there were nine, I think, including things like genomic instability and mitochondrial dysfunction. And then they were updated last year with three others put forward by the same group of authors, although there's been some contention, and there was a separate group of scientists that convened in Denmark shortly before then and put forward some slightly different ones, although there's substantial overlap in the ones that have been added. But the idea behind these hallmarks of aging is that if we dial them up, then we can accelerate the aging process. If we dial them down, then we can decelerate it. And these are conserved across different species. And so in your experiments, you included various different assays to try and understand whether taurine affected the nine hallmarks of aging that were first put forward. And you needn't go into details here because there are plenty of other things I'd like to get to in our short period remaining. But I wonder if you could just highlight the ones that you think might be most important when it comes to the effects of taurine, because we can think of them being like a network in that if you push on one node in the network, you generally affect all the others to some degree. And for that reason, I don't think we should think of them as distinct entities, but rather things that are speaking to each other. But when it comes to taurine, do you think there are some that stand out as likely being particularly important to its beneficial effects?
Yeah, so these hallmarks are, I think, are probably correlate came from the cancer hallmarks, and that is they came up with a list of cellular processes which are affected during aging. So they will keep increasing because these are cellular processes, whether it is a DNA damage, genomic instability, mitochondrial dysfunction, stem cell exhaustion, epigenetic changes, deregulated nutrient sensing. So what is deregulated nutrient sensing for that matter? For somebody who doesn't know about nutrient sensing, they'll say, "What the hell you're talking about?" But what I'm don't mind my language. So basically they are cellular processes. They are all interconnected. All the cells have to function like a machine, right? If you tweak, you are absolutely right. If you tweak something, it is going to have a ripple effect on a variety of different things. When it comes to taurine, it is also affecting all these hallmarks. It is ameliorating many of the dysfunctions happening in cellular processes. But one hallmark that I think is particularly interesting to me personally is mitochondria, because taurine is actively imported in mitochondria, and the one of the known covalent modification of taurine The only known modification of taurine happens in the tRNA. So taurine goes and binds covalently to a tRNA. And this tRNA regulates the production of a one particular protein in mitochondria known as ND6, which is part of the electron transport chain, which is required for generation of ATP in our cells. And in the absence of this modification, the ETC gets compromised and there is a reduced ATP production and more reactive oxygen species production during aging. And if you have mitochondrial dysfunction, it has a cascade of events happening downstream, such as having accumulation of senescent cells or telomerase deficiency induced consequences on DNA damage. So there are all the things are interconnected. So I think mitochondria would is a critical component in taurine mode of action. We are doing now experiments to demonstrate exactly that.
A lot of people describe mitochondria as being the powerhouses of cells, as if they're just involved in generating ATP, but the reality is that they fulfill so many functions. And they're
Absolutely.
really like a family of organelles that are scurrying around cells, moving to different places according to what has to be done. They're involved in building things, redox regulation, so many more things. So they really are fundamental. And the important point is that your experiment suggests that taurine is targeting the underlying biology of aging, the actual processes that are involved in driving the aging process as opposed to mere passengers of it. Although we need to bear in mind the limitations of the hallmarks, one of which is that they weren't systematically identified using some sort of unbiased screen. They were cherry picked based on people's best guesses. But I think for the most part, they are best guesses. Minus some exceptions. I've got a personal bias, which is that circadian dysfunction is clearly a hallmark of aging, in that if you tamper with the circadian system, whether genetically or through apparent light dark cycles or whatever it might be, then you can clearly accelerate aging. What needs to be done though is the gain of function experiment where you improve circadian function and then that extends lifespan. Although I do think that there are some tentative pointers in that direction, probably the best of which was an experiment using automated feeder systems by Joe Takahashi and Victoria Costa Rodriguez that showed that you could further extend the benefits of calorie restriction on lifespan by combining it with time restriction in a smart way, provided that was restricted to the daytime. And when I say daytime, I mean biological daytime. In a roundabout way, that actually brings us to what I wanted to discuss next, which is your upcoming clinical trial looking at taurine and whether it affects humans in similarly beneficial ways to what your experiments have shown in mice and macaques and other animals. And the roundabout way is that I was curious to ask you about whether you might consider looking at some proxy of circadian function. So you could use actimetry, for example, to look at rest activity rhythms, and you could use a measure like interdaily stability to look at the regularity of people's behavioral cycles and whether those are influenced by taurine. But before we get to that, tell us a bit about this clinical trial that you now have going on. It sounds like a heroic effort. I've looked on clinicaltrials.gov and gleaned as much information as I can about it. And I'm sure it will be a very impressive study. There's a lot going on once again. What's your role and what are some of the most important things that people need to know about it?
So before you go into it, you mentioned circadian rhythm.
Mm.
And that is where you need to now appreciate the animal models. Mice do not, most of the mice strains do not produce melatonin.
Yeah, yeah.
C57 does not produce
No,
melatonin
no, it's not
because it
proficient.
has a mutation in the enzyme required to produce melatonin. It
Mm.
is perfectly fine. It has normal day-night cycle. It has no issues whatsoever. Look at us. We have this calcification of pineal gland and we get haywire during aging to really have maintain our proper rhythm. So this is also a species contrast. Any two different species. Now, is it the mice are the good model to do a circadian rhythm experiments? There
Not
are open
melatonin.
questions. I did a lot of research on melatonin earlier, but this is just open questions, right? So one has to use across different model organisms to demonstrate any finding because
Mm.
they may have differences in different species. Okay, leaving that aside, let us go back to the clinical trial. So clinical trial that we have been putting together for almost 3 years. We have been trying to talk with the different investigators, try to figure it out to do the clinical trial. I'm a basic scientist. My role in this clinical trial is basically is and was for many things is to be able to help tweak based on my knowledge that I've acquired over 13, 14 years in doing taurine research to see, okay, where does it work? What are the things to look into it? If you look at muscle function, or if you look at heart function, or if you look at brain function, where and what dosing regimens would be most beneficial to do achieve those aims. But I'm on a back seat in this clinical trial because it is all going to be the credit to the clinicians who are going to do the trial. It is no credit to myself. It is all clinicians who are going to do the work here, and they deserve all the credit and all the things that they will achieve in this humongous effort that we are trying to put it together. As I mentioned before, taurine is a micronutrient which is coming from animal-based diet. And I also alluded briefly that genetics and nutrients have co-evolved. So my hope and my goal here is to do the clinical trial, that this clinical trial will be achieved by clinicians in different populations, which are genetically diverse. So one part of the trial is happening in Germany, where it is already approved. They are recruiting already, and they are going to be testing in a German cohort where female male divided equally is a randomized placebo-controlled clinical trial, and then they will be supplemented with taurine, 4 gram twice, meaning 2 grams twice a day. So that is 4 grams a day for 6 months. Baseline characteristics will be measured at before the trial starts and at the in between for different measurements and at the end. And in this clinical trial, we're looking at different organ system health. And of course, all these different molecular markers like epigenetic aging, molecular aging in the as far as the proteomes are concerned, as far as the DNA damage markers are concerned, all these markers will be looked at, but we are trying to do as robustly as possible. And then we are still in talks in India to do it in vegetarian population, because India being a vegetarian country, I mean, to the most part, a lot of vegetarian population exists in India. So we're going to trying to do it in a vegetarian population in India. In Germany, it's just mostly non-vegetarian population. We are in talks right now to do it in Nepal, which is having a lot of aging population. We are also in talks, still in talks in Singapore. I don't know that how to what extent will precipitate there. And then in the US, we are hoping to do it in African Americans, because then we can get a more closer look at the population, which can tell us now how these different genetic makeups are going to respond to taurine. And that is the stage at which we are. So my role here is nowhere it should be seen. I am only on the back seat. Clinicians are on in the front seat, and they are the one who will do the work, and all the credit is due to them, or it belongs to them.
I think you're being overly humble. Certainly think that you deserve a lot of credit for the work that you've done. I was just looking on clinical trials at the age range. Is it 55 to 75?
Yeah, 55 to 75.
Why the split dose out of curiosity? So two in the morning, two in the evening, and going back to your experiments, one of the things I was curious about was timing, because in both the mice and the macaques, the taurine was given at about 10 a.m., which is the early biological nighttime for mice being nocturnal, and it's the early biological daytime for macaques being diurnal like us. So I wonder if there might be an optimal circadian time, but taurine has a very short half-life. It's
Yes.
cleared pretty quickly. Is that the reason for the split dose? Are you trying to maintain levels above a certain threshold, or is there
Yeah,
something
there are
else at play?
some studies not published yet. Sorry to interrupt you.
Hmm.
So we split the dose for many reasons. We have done some experiments in the mice, a model organism again, where if you can split the dose, it has a better response for organ health, many organ health that we measured. Uh, and the second reason also was, no, okay, this is a model organism. You see better effect in mice if you split the dose. Okay, so what? It's
Hmm.
just an animal model, right? I mentioned to you, and I'm very categorical in stating these are animal models. They are not human beings.
Hmm.
Now, there are evidences in humans where taurine makes people sleep better. These are
Hmm.
layman or empirical evidences. It makes them people sleep better. It having less anxiety. And
Yeah.
third reason is that if you give somebody 4 grams of taurine, you know how much will be 4 grams of taurine? Think about it. How visual it will be visually appealing for you to take 4 grams of taurine in one shot.
I take 4 grams of taurine in one shot, no problem.
No, so you can take it. You can, but it is to comply people to do such job to be for
Yeah.
500 milligram 4 pills given twice a day is much easier to consume.
I actually
And
take it as a powder because it's quite water soluble and it's very
yeah,
neutral tasting.
it,
It's
it's
just slightly sweet, but
I think it is we will learn a lot of information. Yesterday itself, it just so happens, yesterday itself, I was talking to a epidemiologist, and she's a wonderful nutritionist.
mm,
She has a master's in public health and a PhD, and she is the one who is trying to help us put together this clinical trial at the Rutgers Biomedical Health Center. So I
yeah,
moved my lab now to Rutgers University, and one of the reasons to be in a city like New York is that it has a lot of African American population. And when I consider different opportunities available to me, I considered this very seriously because to do monkey intervention, I went up to New Delhi to do the job because I knew how to do monkey research. So being in this environment, it is a clinical center. There is a fantastic opportunities, great investigators available here, and then the large African American population in the city of New York will help us to really get the momentum necessary. I have to be present. You have to be present in the place where the job needs to be done. That's the only way you can get it done. You have to be running around. You have to be running around. If you want to have a benefit of health, you need to be going to a treadmill and exercise. I personally, for example, I exercise. I used to cycle to work on a daily basis, 6 days a week. It helped me. I lost a lot of fat. I'm going to touch 50 very soon. But it, it it can tell you that you need to be present in a place where you want the things to be done. And hopefully, Rutgers University will help us to achieve the aims in African American population present in this locality. Let us hope so, and I'm keeping my fingers crossed that somebody will be interested in, or some people, some clinicians together, they will be interested to work together because we cannot fight aging by working in isolation. We need to work together to work with people who with a diverse backgrounds, different nutrients, different pharma drugs, to put them together, to be able to talk to each other, to tell, hey, this doesn't work. This part of the thing is wrong. This is a criticism of this work. We should have these meetings where we are able to brainstorm. People are promoting a particular molecule. Rather than that, we should be promoting health. Let us put together, right? Is human health, human history is not a history of 70 years of a life of a human being like me, or 80 years, or whatever number of years I'm going to live. It is much longer than that. So we need to keep that in mind, that no matter what we're going to do, we are only on this planet for a very brief period of time in the history of a human being. Let us do something and work together to get it done and make it a positive impact for the children who are going to come after us.
Very well said. And just to mention a few more things about the clinical trial that I was impressed by, one is the use of proteomics, just because now we're seeing these studies looking at proteomic signatures of aging of different organs. And so by using proteomics, maybe you can better understand how taurine is affecting the liver versus the pancreas versus the bone versus the skeletal muscle. And we have a lot of history understanding proteins in the blood. We can tie some of them directly to some of the hallmarks of aging, loss of proteostasis being one of them. And we also know something about some of them being causative in aging. So, for example, there are these parabiosis experiments that tie up the circulations of different animals. And then if you transfer the blood of an old mouse to a young mouse, then it ages faster. And some of that seems to be mediated by protein. So,
That proteomics, the credit goes to Tony Corey, who is on
yeah,
who is
in
going to help
Stanford.
us to do the. He amazing. Is one of the most phenomenal collaborative person I have met in my short history of research. Amazing, very forthcoming. Hey, I'm happy to help. And I think it's all credit of that work will is belongs to Tony. He is to be able to help in such a study and to see what happens. I'm very optimistic. Let us see what happens. We'll know in a couple of years what comes out of it, and hopefully it will be done in many genetic backgrounds of human beings and. We will see what happens, and maybe 3 years down the line, we'll say, okay, this works or this doesn't, or if it works, to what extent. I'm positive that it will work. To what extent it will work, I do not know.
Just before we tie up, I'd just love to highlight the existing clinical trials and the fact that they have shown lots of benefits of taurine supplementation at modest doses. So many of those studies have used doses of 1 to 6 grams. In various populations, there have been a couple of meta-analyses published this year showing an array of different improvements to cardiovascular health, everything from improved left ventricular function, reduced blood pressure, reduced resting heart rate. There was another meta-analysis looking at the components of the metabolic syndrome, finding improved blood lipids, improved fasting blood sugar, again, improved blood pressure. And in some instances, there's evidence of dose response relationships where people who take higher doses of taurine get better responses. And people who accumulate large doses of taurine over time through taking it for longer periods get better responses too. When you couple that with the exercise science research showing that taurine consistently enhances endurance performance, especially in the heat. And there's been some work recently showing that it might well improve thermoregulation by increasing the function of the individual sweat glands too. And I wonder about that in the context of aging as well, because if you think about the current circumstance, then climate change to the nihilists aside, the world is getting hotter, and a lot of older people really struggle in the heat. And so I think that that's a quiet benefit of taurine that might actually be quite substantial in years to come. So there are many publications that are already out there that you can peruse to see the benefits of taurine in people and to understand that it is safe. And that's one of the reasons why I'm personally so optimistic about it relative to many of the other candidates. Plus the fact that one of the intriguing aspects of some of the others, like metformin and probably rapamycin too, is that in some ways they counter aspects of a healthy lifestyle. So this is clearly demonstrated for metformin, which blunts some of the adaptations to exercise training. With rapamycin, it has been shown to offset overload-induced hypertrophy in skeletal muscle in mice. It's also been shown to blunt muscle protein synthetic response to exercise in humans, both of which suggest that if you're taking rapamycin while doing resistance training, you might get worse adaptations to the resistance training. Whereas with taurine, there was some work done on female rugby players recently showing improved power production in, I think it was a Wingate test. So from my perspective, when you couple that with the fact that it's inexpensive, doesn't even taste bad, I am very pro taurine, probably more obviously so than you will be, being a measured scientist and someone who will await the results of the clinical trial that you're involved in. Suffice to say, I'm really grateful for the work that you've done and for your time coming on the show today, because it can be difficult getting scientists to come on shows like this. You've clearly embraced opportunities to speak with regular people. One of the things that I really respect about you is that you do so in a way that's balanced and you don't overhype things. I've heard you say something to the effect of let all the horses run. So while you've invested a lot of time and energy and resources in taurine, if it turns out that there are other candidates that perform better for many people, you will accept that. And it's no skin off your back whatsoever. Thank you very much for your time today. And I just wanted to ask if there's anywhere in particular that you'd like to direct the listeners.
I will direct the listeners to watch this space of research in the healthy longevity area. Lot is happening in this area at the moment, and hopefully in next 5 to 6 years we'll have a more informed, more balanced approach to increase our health. I don't know lifespan because I don't think we'll have any measure of lifespan by then, but to suppress the morbid period that we're going to face at the end of our lives. And I'm personally very optimistic to that is going to happen. To what extent, what molecules, or is a bunch of pills that we might have to take when we are 50, 55 years of age to delay the aging, we will see. Um, and if you can point to people that we are in New York in Rutgers Biomedical Health Center, and so that any clinician or neuroscientist or geriatrician who's interested can get in touch with me. I will really appreciate it because that is what my goal is. And any small help is of a great help. Anything is small. Nothing is. I always believe that you if you have a positive mindset, positive things come around. Positive people with positive mindset come along with you to help achieve the goal. Whole universe will work with you if you think in one direction.
Professor Yadav, thank you very much for your time.
Thank you, Greg. It was pleasure talking to you. And